Okay, so thank you everyone for deciding to listen on my talk on VS-MIS, a vaccine study minimal information standard and its ontological representation and applications. So before we begin, I want to make it clear that the motivation for this project lies in VIO-MIN. So VIO-MIN was founded due to the large amounts of heterogeneous vaccine knowledge and data that exists. It is the largest and most comprehensive vaccine knowledge base, containing over 4,700 vaccines from 216 plus pathogens and non-infectious diseases. All of the information regarding vaccines are integrated using the Vaccine Ontology, but when it comes to vaccine studies, it instead only uses them as a reference. So that means when we're trying to perform some further detail or look for connections with these vaccine studies, VIO-MIN doesn't really have a good way to support this or systematic way to look through all of the details for studies and finding summaries. So ontologically, we have the Vaccine Ontology, which is focused on vaccines, but we also have the Vaccine Investigation Ontology, which is a subset of VO, which does some modeling when it comes to the investigation side for vaccines and some of the initial features, but it's still relatively basic, and we wanted to expand upon this framework. So instead of working on just individual vaccine research studies, we can instead create and design a framework that would be relevant to all vaccine meta-analysis or studies. And we already have a proof of concept based off of an earlier paper focusing on Brucella vaccines, where it was identified 19 specific variables, went to 401 different experimental groups to take a look for which variables that, after we identified and found ontologically, were shown to be significant effectors when it comes to the protection of a vaccine when it comes to the host. So we had this very basic framework where we want to do, but we want to expand on this in order to create part of a minimum information standard. So we have some of these details, but now we want to establish what sort of guidelines that we want to report or summarize when it comes to the vaccine studies. Now, existing databases already use minimum information standard when it comes to adding details. The most famous, I would think, is the Gene Expression Omnibus, which uses Miami and MuSeq data in order to represent gene expression data. And while we can find examples for specific fields like proteomics or for specific immunological assays, like Miata, Misfishi, MyFlow, there isn't anything that specifically focuses on vaccine studies. So, keeping all of that in mind, we wanted to establish a vaccine study minimum information standard, and then make sure that whatever standard that we design would be able to be used to integrate vaccine studies for different meta-analysis. So in order to accomplish that, we had three major tasks. The first is to decide what is going to be the high-level design for the data representation. We need to identify what kind of common metadata types and factors exist that for vaccine studies. We need to develop use cases for both an individual vaccine study and also when we're dealing with vaccine meta-analysis. So when it comes to the high-level design, we looked at what was done for the meta-analysis, but we also focused on the ISO framework and also with imports data model. So the ISO framework is this nice generic model design, which we thought it would be relatively simple to adapt. But we noticed that when going through the different protocols and measurements and technology for what's going on, we did not have a nice and concise area where the actual experiment is being modeled. All of those components are spread throughout the different levels when we tried applying it to vaccine studies. So, and then we went over to, okay, another place where we get a lot of vaccine studies is from import, which is focused on the different immune expression responses as part of the experimental procedure. And while it has a lot of nice details, we still lack into issues with some standardization on what's being reported and where. In addition, import also makes a difference between unplanned visits of a person going to an experiment and collecting a sample there, which is then separated out from an actual experimental sample generated from the experiment, which we thought was could be consolidated when it comes to the actual vaccine studies. So keeping both those in mind, we wanted to propose the C framework, where taking those two ideas, we just wanted to explicitly add an experiment level step, which is part of the actual study, and that is the assay is part of. This can either be something that shows up clinically or as part of the lab model, so they can either be collected through an experimental lab model or collected from a clinical biosample. These assays we wanted to focus on will generate some sort of results data, and that whatever experimental factors that we think or that would likely have some effect, either as part of research question or known from prior knowledge, these experimental factors would be listed as part of the experimental step. Now, when we talk about the idea of variables, we have the focus of both the independent variables and the actual dependent variables, which ended up being confusing since we're using the word variable in multiple different ways. So to try to elucidate these relationships and be more clear, we worked on this data reality variable model. So at the most basic level, and I'm sure most of you are familiar with, we have either data or metadata, which reflect some entity in reality. This is something that we can represent ontologically as either an entity or a specific quality. And this is what shows up in the different BFO-aligned ontologies. Statistical variables, which are an information content entity which are used as part of statistical analysis, can either be about an independent variable or a dependent variable, depending on if it's being used as part of the inputs of the data or if it's being used to describe the entity in reality. So in this case, we wanted to provide clear framework that when we're talking about this ontologically, we can use this in order to make more sense when trying to find the underlying mechanisms that occur between the data and what's going on in reality. So that's very nice and abstract, but what does that mean in practical notation or as part of the minimal standards? Because in many cases, when we're recording information about practical data annotation, we can probably just use the actual entity in reality in terms of the ontological ID. So when talking about biological sex, even if we have a whole variety of different biological sex data, which corresponds to some entity, we would instead play a note that it's going to be about or just record this is biological sex quality. And so, which raises a question. What context would we use the separation and why does this matter? So when it comes to the realm of semantic ontologies or trying to find some of the relationships with what's set up, all that would be about the entity in reality or the qualities where we're looking at these axioms and details. When we're trying to understand the mechanistic modeling of a vaccine study, then we're interested in both the actual variables, which can serve as inputs or data items for what goes in, but also the different entity in reality they reflect. But then when we get over to something when it comes to using inputs from machine learning AI algorithm or trying to make predictions, then strictly speaking, we're only interested about the data, especially when we go into larger models that black box or set up some different weights of the data in order to find some underlying pattern or perfect dimension reduction. So keeping that all in mind, following the Assay framework, we end up with this data-oriented model. The main changes are the inclusion of the experimental step and the inclusion of two different types of studies, that of host response to vaccine and that of vaccine challenge studies. So host immune response to vaccination is the first step. You get the vaccine in the host, immunize them, then observe the response. If you then add the pathogen to a study and then track the survival of either the host or the pathogen, then you end up with the actual vaccine challenge study. And again, want to reiterate that the use of experiment covers both clinical visits or that tend to be more human-related, but also more lab model experiments with animals that link up to the different factors. So when we take a look at this more realism-based approach of the different qualities and entities. We have a series of different processes in red. We have different material entities that are generated in blue. And then in the burgundy colors, we have the different related factors that we found to be consistent in reporting across the different vaccine studies. I'm going to go more in depth later, but this is just a visualization of how it will actually look. So when we get into the metadata representation within the C framework, we'll first start at the S level or study level. And this is the information where we tend to give a listing of the different study names, the identifiers, the descriptors, all of the useful metadata that allows us to quickly find information or link it together. We do have a note that we have about 20 experimental factors, which are being listed as, hey, what kind of metadata or factors that this experiment was interested in looking in order to help that. And I put a small asterisk because we identified about 20 different factors, and which we grouped into 5 categories. So these are those relating to the material entities, the vaccine, the pathogen, and the host, and also with the processes of vaccination and challenge. Now, right now, the vaccine and pathogen representation can be simplified to only one term, simply because we have well annotated those terms as part of vaccine ontology. And since that means we could group the vaccination process, the challenge process for the different intervals, and then we have the host metadata, which is the biological factors which are relevant to both the vaccination and as part of the challenge step. In these cases, whatever factors are mentioned on the study level are going to correspond to the different variables and data that the study are about. Then when we get into the assay level, we're getting records using pre-existing minimal information standards, but we're mostly focused on reporting the assay types and other time measurements. Now, assays can vary depending on what kind of vaccine study we focus on. Immune response challenges tend to focus on host immunogenic response rates, so stuff like ELISA, flow cytometry, microarray data, while challenge studies focusing on survival would be stuff like the host survival assay, the pathogen colony-forming unit reduction assay, or the gut colonization. The third category, which can occur to both of these, when we include data analysis tools as a type of assay. So this involves getting some form of raw data and then using a statistical transformation in order to find some sort of pattern or to have them be realized in order to identify what's going on. This can be either used to find like false discovery rate to see if something's significant or not, or we can do something more complex like turning gene expression data into gene pathway data, which you can see in web tools like David. So when we get into the actual modeling and generation of new terms, currently within Vaccine Ontology, there's about 104 related terms when it comes to the different study design variables, which are the official term used for the variables. Also with the different data that's being recorded as measurement data within VIO. We added about 70 new terms into Vaccine Ontology as of this moment. And then we again have this modeling depending on if we want the design variable to focus on the real-world entities or entities in reality, the actual data and metadata. I want to point out that when we're talking about the independent variables, that's when we have the variables focusing more on the data item about a real-world entity. And then when we're talking about the dependent variable, that's when we see it's about whatever entity in reality, as it's used as a classification or categorization of the statistical analysis. So we have all those words. What does this look like in practicality? So we have each of the different properties mapped to a specific ontology ID. In most cases, I try to find the appropriate class level identification, but for some of the names and experiments, instead went with the annotation property that we would use within the different OEO boundary ontologies. We give it a name if it's not one that's actually split. We have a note on what sort of experimental variable, experimental factor. Is this a control? Is this following vaccination effect? What vaccine is used? Is there some sort of intervention? And then try to keep that consistent. When it comes to the different intervals, we include both the unit and the categorical data in order to keep things consistent. when we take a look at what happens on the host species or the details, we make a note if we know if there's a change in the actual metadata. So host comorbidity no means that there's no mutations or disposition that would cause a negative effect on the organism. Host disease state no means that the organism is healthy. If we get in a case where age is not specifically mentioned as part of the study, which mostly occurs in older studies, we say that it's unlisted in order to distinguish between we know it's healthy or we don't know, or if it's not being reported. Now, when we go over to the assay side, we identify using the same sample name and sample ID to link them together. And then we provide details on the technology type for the actual assay being used. If there's any other procedure or protocol that's mentioned, what sort of material that it was collected as part of the sample, how's the data being recorded. If we have multiple results in the schema, we report it as a vector. Now, I did a more simplistic example when it comes to what happens when we want to look at meta-analysis or multiple samples or multiple studies. In each of those cases, when we're taking multiple different samples and then trying to perform some form of statistical analysis on them, it's going to be the same even if it's across 10 studies or if you're just doing a comparison across 2 different time series or time points. So in the example of human gene expression due to an influenza vaccine, we see that, hey, there's a microarray, we use Affymetrix as a platform, it's collected from peripheral blood mononuclear cells, and for the day 3 and day 7, we're given like a matrix of the assay result of what's the change. However, when we go over to the actual human gene expression for what's going on, for the technology type, instead of talking about the microarray or how it's collected with the platform, we say that this is undergoing a statistical analysis. And that moreover, the actual platform that's being used is whatever statistical hypothesis tool that we use to test it. In this case, it would be a d-test. And then from there, we would get a list of whichever cells which were shown to be significant. Incidentally, that's why gene 1 and gene 2 in the third column doesn't correspond to the ratio for gene 1 and gene 2. They're technically speaking different gene names. So in summary, we've developed the C model based off of ISA framework, the import share data model, and our prior work with the Brucella vaccine variable meta-analysis. In order to make clear the ontological representation of what we're talking about, we developed the data reality variable modeling in order to make sense of this and to support some data analysis. Currently, the vaccine study minimal information standard uses both the C and DRV models, where we have about 104 general metadata and variable classes in VO. We've identified 20 specific vaccine variables as relevant. We've also provided an example of the Brucella and flu vaccine study demos of this. As for future work, we have this framework. We still need to get this submitted by and peer review to make sure that our thinking is completely correct and no further modifications. But we want to use this in order to provide a general update to all the different vaccine studies and work we've done in VIO. Moreover, our framework here is only focused on traditional use of vaccines to immunize against a disease. Certain vaccines, such as the cancer vaccines, are used to as an intervention for cancer treatments, and so would require a different set of modeling and details, which would end up being more complex. We also want to expand upon these models for just vaccine studies and see if we can line this work as part of the NI data project. And we just want to acknowledge our funder, our program officer, and also my department head who provided some commentary that we've incorporated as part of a new paper, but didn't incorporate when we submitted this for VDOS. And thank you.